So continuing on, we talked about primary structure. The next form of structure that we need to discuss is then secondary structure. So in a secondary structure, we have local structure. So this is just getting one step bigger. So it's not necessarily just how they're in order, but it's how they interact with each other in a local way. So just right next to each other. So local structure due to hydrogen bonding. So that's the big ticket right there. What you need to remember, hydrogen bonding is what causes secondary structures. So local structure due to hydrogen bonding between neighboring amino acids or peptide chains. So there's particular ways that this will look. The first one is called an alpha helix. So this looks like a spiral, like a ribbon. That's how they will actually draw it in like the cartoon versions of peptides that they draw, is it'll look like a ribbon or a spiral. These are called alpha helixes. It's a little hard to tell what's going on in the actual molecule 3D representation here on the left, but what this is showing is that you have, let's keep the yellow, the carbonyl from one peptide, one amino acid, has a hydrogen bond with the NH. So hydrogen bond in red or pink, whatever you decide that color is. So the carbonyl from one amino acid interacts with the NH from a different amino acid on the same chain. So this is hydrogen bonding in the backbone between the carbonyl and the NH. So that's really important. This is between the backbone of the same peptide chain, the same protein chain. These alpha helices are always turning right-handed. So the direction of the spin is always the same. And it's almost always 3.6 amino acids per turn. So it takes 3.6 amino acids to go 360 degrees around. And keep doing that. So the hydrogen bonds are between I and I+4. So between, like if I is representing amino acid number 1, then you have a bond between amino acid number 1 and amino acid number 5, because that's 4 greater than that. Or if you have amino acid number 5, that is going to hydrogen bond with amino acid number 9 in the alpha helix. Okay, R groups. So this is also super important because this is how you'll see some questions about this, is what do the R groups look like, or predict based off of the amino acids that I give you what the secondary structure is going to look like. So in this case, R groups point out of the helix. So they stick out. So let's find some here in our example. Well, they use tiny R groups. These purple groups here are the R groups. And you see that they all poke out to the side, right? They're all on the very outside poking out. So because of this, you usually have large R groups. So things that are really big like to point out to the side instead of jamming into other things, right? So if you have large peptides next to, or large amino acids next to each other, it will often form an alpha helix. The things that will break this really easily, so as soon as you hit one of these, it's going to break apart this chain, are glycine and proline. So if you see a proline in a chain, it's not going to have an alpha helix after that point. Or it could have maybe 2 alpha helices next to each other, but there's a kink in the system. So it's like a ribbon one way, kink, and then another ribbon. The next one is called a beta pleated sheet. So we had alpha, now we have beta. So this is a similar interaction in which it's backbone. So it's the same carbonyl hydrogen bonding to the NH. However, this time it's a different orientation. So instead of a spiral to have that those backbones interacting, you have a zigzag. So it's not just I+4 number of amino acids away. You have this zigzag. So it's going to be different based off of what structures you're talking about. So individual strands weave back and forth to form a sheet. So you just go back and forth and back and forth. It's called a beta sheet. beta pleated sheet. They also will not be 100% flat. They'll be a little bit cockeyed, same as like how we draw our structures. So that's why you see the like cartoon behind it as kind of a zigzag pleated piece of paper. It's the same, that's what it looks like once you actually draw it or make a model of it. That's what it actually looks like. And then again, we talked about what R groups are important. So I'm going to highlight some of these R groups here. Some of them go behind, so you can't really see them, but you see, if you look at this in 3D, these R groups are coming out towards you or back into the plane of the paper. So like, for example, there's one here, but it's behind that white one, so it's hard to see. Okay, so R groups, because they point above and below the sheet, they typically are small, trying to get out of the way. So R groups are typically small, they point up and point down. And then additionally, these have very high strength. These are high strength interactions. So you have lots of them, kind of makes like a piece of paper. So it's, if you pull on the ends of a piece of paper, it's not going to unravel very easily. So this is the primary interaction in silk. So you can imagine the strength of silk is from these beta pleated sheets. The last one is called a triple helix. Why it's not a gamma triple helix, don't quite know, but that's just me. These are when 3 polypeptide chains weave like a braid. They are very strong. We don't have necessarily a good predictor in terms of the R groups for what will predict for these to happen, but these are the strongest form of our secondary structure. So for example, collagen, which is 1/3 of all protein in vertebrates, it's our connective tissue between like our skin and our other organs and everything. It's our blood vessels, our skin, our tendons, our ligaments, cornea in our eye, our cartilage, all of the very structural things in our bodies are due to collagen, which is made or has this triple helix structure. So for example, just this little what collagen is used for, it keeps your skin elastic. And so what's something that's really important to that is vitamin C. So vitamin C has a lot of the building blocks for collagen. So natural collagen production requires vitamin C as a nutrient. And then the reason that we age, or well, we age because of time, but the reason our skin ages and we lose elasticity in our skin is because over time, this triple helix in our collagen in our skin loses integrity. So if you think about like a rope, over time, it's just going to get a little bit more frayed. These amino acids are going to, or peptide chains are going to get damaged just over time. And that's why we see aging in our body.